Here we can see what a large organ the liver is. This is a cadaver shot, and here we can see the liver, the stomach, the greater omentum coming off of the greater curvature of the stomach, the gallbladder. This is the small intestine. In this closer view of the liver, we can see the two major lobes of the liver, which are simply called the right lobe and the left lobe. We can also see the gallbladder. Here we're looking at the inferior or bottom surface of the liver, and we can see where the inferior vena cava was with this dark collapsed area. This is the gallbladder. Those two landmarks help us identify the two minor lobes of the liver. This is the caudate lobe, which is posterior. In other words, it's next to the inferior vena cava, which is in the back. And this is the quadrate lobe, which is adjacent to the gallbladder. It's anterior. The organization of the liver is somewhat like the lungs in a sense, where the lungs had lobes that we could grossly see. So does the liver, the two major lobes and two minor lobes. But all of those lobes look the same at the microscopic level. They're all made out of a basic subunit called a lobule. Here you can see the arrangement of lobules. It's very geometric. Lobules are cylindrical subunits. So this is a cross-section through a number of lobules. This is a higher magnification of a cross-section through a single lobule. That hollow center is where the central vein is located. So there is a central vein inside of each lobule, and then the cells that make up the lobule, the liver cells, are called hepatocytes, and the arrangement of the hepatocytes are that they radiate out from that central vein. Here's a diagram of a single lobule so we can see a few more of these characteristics. We can see a little bit of the cylindrical nature of it. We can see that the blood is going to enter from the outer periphery of the lobule, and the blood is going to flow through very leaky capillaries called sinusoids towards the central vein. We can also see the hepatocytes that are radiating out from the central vein. So let's start to consider how the liver does its job. The basic premise is that when blood flows into the liver, that blood is going to flow through the lobules. It's going to enter at the outer periphery of the lobule, and then the blood is going to flow through a very leaky capillary called a hepatic sinusoid towards the central vein. As the blood is moving through the hepatic sinusoid towards the central vein, this is when the hepatocytes do the job of the liver and process that blood in the hepatic sinusoid. So as far as, the numerous jobs that the liver does, some of those we've already talked about. For instance, remember that the plasma contains proteins like albumin and angiotensinogen. Where do those proteins come from? Over half of the plasma proteins are produced in the liver. The presence of the sinusoid, the leaky capillary, permits entry of bigger structures like protein. Another job that we know the liver does is the liver removes dead and damaged red blood cells from the circulation. There are specialized macrophages that live in the liver. They're called Kupffer cells. They're, a type of hepatic cell, so to speak, although they're derived from a white blood cell. And as the blood flows through the hepatic sinusoids, any dead or damaged red blood cell is phagocytized by the Kupffer cell and taken out of circulation. You might recall that the liver produces glycogen, which is the storage form of glucose. So when we're in a fed state and when insulin is present, that would promote the storage of glucose in the form of glycogen. The hepatocytes do that job. And the hepatocytes can also generate glucose. So under the influence of glucagon, the liver can actually use amino acids and fatty acids, non-sugar ingredients, to produce new glucose in a process called gluconeogenesis. So those are the jobs I think you're familiar with already as far as what the liver does. And here's a few new ones for you. One of the sources of blood flow to the liver is blood that has just flowed through the small intestine and therefore received the nutrients that entered in from the GI tract. And so taking a look at the outer periphery of the lobule, let's see where the blood is coming from. There are two sources. One is the hepatic artery. The hepatic artery is bringing clean, oxygenated blood to the liver. And then the other source is the hepatic portal vein. The hepatic portal vein is part of a portal system. Remember, a portal system is one capillary bed that leads into a second capillary bed. The first capillary bed is in the small intestine, where it receives absorbed nutrients, and then that blood is shuttled directly to the liver through the hepatic portal vein. The hepatic portal vein contains all of that blood from the small intestine, which the liver will clean up, so to speak. It will metabolize protein, carbohydrate, and fat present in the blood. The liver will also further process this blood coming in from the small intestine through the hepatic portal vein by detoxifying and purifying the blood, and it does that by metabolizing chemicals, drugs, alcohol, and anything other than nutrients that is present in the blood. In addition to all of these jobs, the liver also produces bile. And so I want to take a closer look at what that means. What is bile? Well, when we think about some of the jobs that we just talked about, you can imagine that the liver is going to really start to amass waste products, because we said that the liver removes dead and damaged red blood cells from circulation. That would be quite a bit of waste. And it's also metabolizing substances. And because of this, it generates a lot of waste. So what do the hepatocytes do with that waste? There are small canals that are in contact with all the hepatocytes called bile canaliculi. And the bile canaliculi function a little bit like a garbage disposal, in that the hepatocytes that remove substances from the blood, what do you do with them? You put that waste product in the bile canaliculi. That makes up a large percentage of what bile is. Okay, so when these wastes enter into the bile canaliculi, they accumulate there and they flow to the outer periphery of the lobule into a bile duct. All of the bile ducts on the outer peripheries of all of the lobules ultimately merge together. Ultimately, the bile ducts surrounding the lobules merge into two big ducts that leave the liver, the right hepatic duct and the left hepatic duct. Those two ducts merge to form the common hepatic duct. The common hepatic duct continues on as the common bile duct to the small intestine, okay, to the first region of the small intestine, the duodenum, where the opening of that duct is controlled by the hepatopancreatic sphincter. Usually that sphincter is closed. If it's open, bile will enter into the duodenum. If it's closed, bile won't enter into the duodenum. But I'm going to consider the composition of bile, the function of bile, and the entry of bile into the small intestine in the next video about the gallbladder.